All right, so here we are to have a hopefully quick video on protein folding. There are 4 levels of protein folding: primary, and this is just the abbreviation for primary, secondary, tertiary, and quaternary. All right, with primary, this is what happens during translation. So if you think about proteins and how they're made, DNA is transcribed into RNA in the nucleus. That RNA leaves the nucleus, goes to the ribosome, and that ribosome then translates that RNA into protein, one amino acid at a time. So it basically takes an amino acid, okay, AA, and it binds another amino acid. And then it binds another amino acid. And then it binds another amino acid. Okay? And so it just basically keeps making this long chain. Most proteins are hundreds of amino acids long. So even though I'm just drawing 4, just know that that's an abbreviation for sure. The type of bond between these is called a peptide bond. It is just a covalent bond, but we call it peptide. Sometimes you will hear proteins called polypeptides because of the presence of those peptide bonds. All right, secondary structure is when this chain gets long enough, it will start to coil and fold in on itself, and it forms 2 major structures. It forms an alpha helix, so alpha is the Greek letter. Kind of looks like an old-timey telephone cord, or it can form a beta pleated sheet, and that's not a very pretty beta pleated sheet, but basically if you've ever folded paper back and forth when you were younger and made a fan to fan yourself with, that's kind of what a beta pleated sheet looks like. And by and large, these are due to hydrogen bonds between the hydrogens that are sticking off all over the amino acids. Because if you remember, I'm drawing an amino acid very simple here, but it is a central carbon with a hydrogen sticking off the top, an amino group sticking off to the left, which has 2 hydrogens, a carboxyl group sticking off to the right, which has a hydrogen, and then an R group coming off the bottom, which may or may not have hydrogens. So all of those hydrogens are kind of interacting, and that causes this molecule to start to form these shapes, and that's secondary structure. Tertiary structure is then when the R groups or the functional groups start to interact, and we start to see the molecule twist and form more in on itself. And so you'll see, and this is going to look like a big hairball when I'm done, so I'm going to try to draw it where you can see as I'm going. So you might have an alpha helix, and then a beta pleated sheet, and then another alpha helix, and then another beta pleated sheet, and then another alpha helix, and then another beta pleated sheet, and you see it just starts to look like a big hot mess. So this is due to interactions between the functional groups. Functional groups. And so what you're going to see here is you're going to see that your positive charges are going to be attracted to your negative charges. Okay, because opposites attract. So anywhere you have a positively charged functional group, it's going to be seeking out a negatively charged functional group. Or a negative is going to seek out a positive. Anywhere that you have nonpolar or hydrophobic amino acids, they're going to try to get into the center of this protein, because most proteins are in an aqueous solution, whether that's your cytoplasm or your blood or just out in the environment, whatever. And so they want to get in here where it's safe, where there's no water, because they are nonpolar and they don't like water. You might also have some polar functional groups. They are hydrophilic. They love water. So they're going to try to move more to the outside where they can interact with that water. And then you might have some sulfur. Cysteine has a sulfur. Sulfur is going to always seek out another sulfur and form this disulfide bridge. And that is just going to be a very strong interaction between 2 sulfurs it's going to be really a really strong bond, and it's going to give the molecule some stability. All right, and then there's also some hydrogen bonding that's going on here too. Hydrogen bonds. So even though that's what's happening here, there's also some hydrogen bonds between functional groups. And this is really important that this guy gets folded correctly, because we know that structure leads to function. So if it's not folded correctly, it's not going to do the right job. And that's actually what's going on with a lot of genetic disorders. There's a mutation in the DNA, which means there's a change in this chain, which means there's a change here, and there's a change here, and potentially here. And so when they're not shaped right, they're not going to function correctly. Quaternary structure is not all proteins make it to this. Some of them are done right here. They are tertiary and they go and do their job. But some of them need to join up with another peptide chain. So we call this a peptide chain. When it gets down to its final state, it's a protein. So if this is its final state, that's a protein. But for some of them, it's not. Some of them have to join with another tertiary, so another one over here. They're going to come together and form a quaternary. And hemoglobin is a good example of this. Hemoglobin has actually 4 of these chains. So 4, you have 2 alphas and 2 betas, not to be confused with these alphas and betas. But you have 2 chains coming from 2 separate pieces of DNA that are going to fold up. And so what you're going to see is if those are the alphas and these are the betas, and I'm just drawing them kind of like a big hairball, but imagine they're more like this. Those 4 have to come together for this to be functional, for this hemoglobin to be functional. So hemoglobin is an example of a protein that does have a quaternary structure, but there are others, like I said, that don't. There are others that this is where they end. So not all of them have that, and there's just a bunch of different intermolecular forces that go into holding these guys together. All right, the other couple of things we needed to talk about were chaperonins, and chaperonins help the protein fold. So they are literally chaperoning that process of folding, and I shouldn't just say they help it fold correctly, because if it were to fold incorrectly, again, structure would not be correct and the function would not be correct. So chaperonins come in. We'll talk about those more in Unit 6 when we talk about transcription and translation and what happens to proteins after that. But that's what they're going to do. All right, denaturation. Denaturation is a process. You may hear it as a verb, to denature, or you may hear that the protein has been denatured. So it's an adjective, so we can use it all kinds of ways. But when you think about the nature of something, like the nature of a person, it's kind of their personality and how they work, how they live life. With a protein, their nature is due to their function and their structure. So when we talk about the nature of a protein, it is the structure of that protein and therefore how it functions. So to denature it is to take that away. And so what we see happening is if we have this protein, let's say that we have a very simple protein that just does that. So you got 2 alpha helices and a beta pleated sheet and it folds in on itself. When it gets denatured, it's basically going to unfold, and it may even unfold back to primary structure. And so now that it no longer is shaped this way, it cannot function. Things that can do this are pH changes. So for example, the enzymes in your stomach work great at a pH of 1 to 2. If you put them in a nice safe pH of 7, they literally will unfold and they can't function. If you take enzymes from your blood where it has a pH of 8 and you put them in your stomach, they can't handle that pH. They're going to unfold. Temperature is another one. This is why fevers can be so dangerous and why we want to keep them from getting too high, especially in adults. Children seem to be a little more flexible, but adults not so much. If your temperature goes too high, it can cause your enzymes, your proteins, to unfold and they won't function. And so they're not doing all the jobs that you need them to do, and therefore you can have some real problems with your body. It can actually lead to death in severe cases. And then salinity or salt content is another thing. So you can't take an enzyme that's used to being in a real salty environment like the ocean and put it in fresh water, because it will fall apart and it will not function anymore. And that is all because the structure of a compound is related to its function. We can look at this in big cases like your feet. You have flat feet because you are bipedal. We don't climb through the trees for a living, so we don't need opposable big toes like our thumbs. We need flat feet because we walk on 2 feet. So that's kind of a big example, but you can take it all the way down to molecules, and if they are not in the right structure, they will not function. And while we've been talking about proteins today, this is true for anything. This is true for carbs, it's true for lipids, it's true for nucleic acids. If the structure is not correct, the function will not be correct either. And so that can be really harmful all the way up to lethal. And so that, like I said, this is something I'm 99% sure you can use this on the AP exam somewhere. Every year my students have been able to include this concept in one of the free responses and earn a point. So keep that in mind. I will talk about it all year long. And that is that. I hope that's helpful. If you have any more questions, please let me know and we can go over any of this again.